So this is the third installment of the Lymph and Immune System lecture. And we're going to start this PowerPoint by just finishing up the cell types that are part of the immune system response. There's one more cell type that we have left to cover, and that's the NK cell. Now, if you recall, we talked about this cell in a nonspecific second line of defense. And these NK cells are nonspecific because they simply go around looking for any type of antigen they don't recognize, and they go and kill it. But how do they kill it? So here is the mechanism for how the NK cells actually work. Let's say this is your enemy cell, and this is your NK immune cell. This NK immune cell, once it finds an enemy cell it doesn't recognize, it's going to attach to it, and it's going to secrete what's called perforins. These are proteins, and these perforins, if you can look closely, they create pores in the membrane of the enemy cell. And through those pores, the NK cell then secretes something called a granzyme. And a granzyme is a type of enzyme, and this is an enzyme that induces apoptosis. Now, if you recall from cell biology, apoptosis is the process of essentially cell suicide. So these apoptotic enzymes will go into the cell, specifically to the nucleus, and it'll tell the cell to commit suicide from the inside out. So the cell itself starts degrading, and it eventually disintegrates, and a new cell called a macrophage, which is very, very large, will come. It'll notice the degraded enemy cell, and it'll do phagocytosis and eat it. So these NK cells, I think they're one of my favorite immune cells because their function is just so cool. And it's just such a umbrella kind of function where it can do this with any kind of bacterial or foreign cell. So secrete the perforins, make the pore, to then secrete the granzymes to go in through the pore to signal cell suicide. So interferons are actually pretty cool. When a cell gets infected by a virus, that infected cell is going to secrete a chemical called an interferon. And I'm actually going to use my pen here to show you the cell. And here is the virus that has infected that cell. And when this virus infects this cell, the cell knows it. And the cell is going to secrete chemicals to the neighboring cells around it. This is my way of showing you chemicals that are secreted. These secreted chemicals that come from the infected cell are called interferons, and they go to neighboring cells, and they bind to receptors on this neighboring cell to send a signal internally to say, hey, I've been infected by a virus, and this cell will then create a protective mechanism, and they create these things called antiviral proteins. Now, I didn't put that on this PowerPoint, but I know a lot of you always ask me lots of questions on the extra details because it brings everything full circle, which is wonderful. So when the interferon binds to the receptor, it sends an internal second messenger signal, and the nucleus is signaled to create antiviral proteins. And these antiviral proteins essentially protect this cell from getting infected from viruses. These interferons will also go and they will send a signal to these large cells called macrophages. And when the interferon goes to the macrophage, again, it binds to the receptor on the membrane of the macrophage. And it says, hey, macrophage. It sends a secondary messenger signal, and that macrophage gets signaled to then go and engulf all virally infected cells. So 2 big jobs of the interferon: to warn neighboring cells to protect themselves, and to signal macrophages to come and do phagocytosis of the infected cells before this cell can burst and release the viruses everywhere. Now, the next system is kind of a trickier one. It is pretty cool, but there's a good amount to learn on this, so I'm going to go through it very slowly, and I'm going to tell you exactly what I want you to know from this for the test. This system is called the complement system. And it's called the complement system because the proteins that are used to run this system are called complement proteins. And these are proteins that are made by the liver, and they get activated and secreted in the presence of a pathogen. So what you're seeing here is a cell membrane inside the cell, outside the cell. This could be a bacterial cell, let's say. The complement proteins start with letter C and they have numbers next to them. So these complement proteins, if you notice, they kind of build on top of each other to create this whole complement complex. And we're going to talk about what the function is of these complement complexes. We'll first talk about 3 ways of activation of complement, and then we'll talk about the methods that complement uses to actually destroy pathogens. 3 ways of activation: classical pathway, alternative pathway, and lectin pathway. 3 methods of actual functioning of the complement pathway to kill pathogens is through activation of inflammation, doing something called immune clearance, doing phagocytosis, or doing cytolysis, which I also call the MAC attack. Make sure you know everything on this slide. I'm going to go through each one of these bullet points and I'm going to explain them to you. But this is your big outline of what this entire complement system is about. So, activation of complement happens with 3 pathways. The first pathway is the classical pathway, and the key term here is that it requires an antibody. So, antibodies are required to get this classical pathway started. When your body and your immune system notice a pathogen, it oftentimes creates antibodies against that pathogen. So let's say we're talking about a B cell. When a B cell notices a pathogen, it turns into a plasma cell. And the plasma cell makes antibodies. And those antibodies go to the bacteria and they come and bind to the bacterial antigen. This antigen-antibody complex, if you recall from the last PowerPoint, sends off an alarm-like signal. We're just going to put that alarm signal onto it. So this antigen-antibody complex is written here as AG-AB complex. The classical pathway of activating the complement system requires this antigen-antibody complex. When you make an antigen-antibody complex, this antibody here will change its shape slightly to expose complement binding sites. So let's say initially your bacteria looked like this, and it had the antigen here, and here was the antibody that was coming to bind. Eventually, after binding, here's the bacteria again, here's the antigen, and now here is the antibody. If you notice, here it didn't have a binding site, and here it now has an exposed binding site. This binding site is called the complement binding site, and it binds to this protein called the C1 complement protein. And the C1 complement protein will come bind here, and when C1 binds, it sets off a domino effect, almost like a cascade, where other complement proteins will also bind. Eventually, all of these complement proteins will all come and bind into this large complex, and they can oftentimes start binding directly to the cell membrane. So when complement proteins start binding directly to the cell membrane of the bacteria as well, they can build what's called a MAC complex, membrane attack complex. Now, we haven't talked about this yet, but if I go back a slide or two, this right here, this is a MAC complex. If you notice, you have a series of complement proteins binding into the membrane, and it creates a hole. Now, one hole in a membrane is fine, but what if these complement proteins created 1,000 holes in the cell membrane? So we've got a hole here, we've got a hole here, we've got a hole here, 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 here. With all of these holes, you're going to lose the integrity of the cell membrane, and it's eventually going to just disintegrate and fall apart. This is very good for you because if the membrane of the bacteria falls apart, then the bacteria dies. So because you had an antigen-antibody complex, you were able to expose the C1 binding site on the antibody. When C1 binds, it creates a cascade of all other complement proteins binding as well. Eventually, those complement proteins can bind into the cell membrane, creating the MAC complex. If you make enough MAC complexes, you are going to destroy the integrity of the bacterial cell membrane, and it's going to fall apart, and that bacteria will die. The alternative pathway does not require an antibody. What you have here are these complement proteins called C3 proteins. The C3 protein will break down into the blood into C3a and C3b. So right now, all of you healthy individuals have C3a and C3b in your blood, swimming around in the plasma. If you by chance have a tumor cell, a virus, bacteria, or yeast that get into the blood, the C3b protein binds to it. When C3b binds to pathogens, it's going to trigger another cascade reaction where you're going to have a lot more C3b binding to the actual bacteria. So here's a bacteria, and imagine you have a ton of C3b complement proteins binding to it. Now, one of the effects of binding a ton of these C3b proteins is to build a membrane attack complex. In a few slides, I'm going to tell you something else that happens when you have a ton of C3b binding to the membrane of a bacteria. But for now, again, you're able to create a MAC complex to create pores in the membrane of the bacteria and disintegrate it and kill the bacteria. The last pathway is called the lectin pathway, and the lectin pathway uses lectin proteins. So lectin proteins are proteins that just swim around in your blood plasma. When you have a bacteria, oftentimes bacteria will have these carbohydrates sticking out of the membrane of it. So sugars. The lectins in the blood can come and bind directly to these particular carbohydrates. When a lectin, and I'll label it L here, comes and binds to the bacterial carbohydrate, this attracts C3 proteins. So we're going to set off a cascade of C3 proteins, and you know that in the blood, C3 breaks down into C3a and C3b. So now, because the lectin bound to the carbohydrate, it activated the C3 proteins to break down into C3a and C3b. You can coat this cell in C3b again, and it can destroy the cell membrane. Now, this is where we add a little bit more detail, and you're going to hear me talking about C3a and C3b a little bit more. And there's a good number of questions on the tests that ask you what the function is of C3a and C3b. So just be sure that you're learning them and you're distinguishing them from one another. So when you activate complement, whether it's through an antibody, whether it's through lectin, whether it's through no antibody at all, the mechanisms of destruction are 4 of them. The first one is inflammation. Now, inflammation, if you've ever bitten off a hangnail before and it peels the skin back, your finger ends up throbbing, it turns kind of red, it swells up a little bit, and it gets very warm. That whole process is inflammation. The point of doing all of those things is to help kill bacteria. So, in inflammation, the C3a proteins, and if you recall, I'll write this down one more time just to keep it consistent. C3 breaks down into C3a. C3b. C3b, we talked about it coating all bacterial cell membranes and creating MAC complexes. C3a we haven't talked about yet until now. So the function of C3a is to stimulate these cells called mast cells and basophil cells. We talked about basophil cells in the last PowerPoint, but we haven't talked about mast cells yet. C3a is going to tell both of these immune system cells to secrete histamine. The histamine is going to elicit a cascade of events that's going to cause inflammation. So because you activated C3, C3 broke down into C3a and C3b. C3b is able to bind to bacterial membranes to create the MAC complexes. C3a is able to activate mast cells and basophils to secrete histamine, and this is the chemical that actually begins inflammation. So inflammation and histamines can activate and attract neutrophils and macrophages. Remember, both of these directly attack bacteria. And all of this will help destroy pathogens. So because we were able to activate C3 in the presence of bacteria, C3 broke down into C3a and C3b. C3b is able to coat bacterial membranes to cause MAC complexes. C3a is able to activate immune cells to secrete histamine to begin the inflammation process. Again, the whole point of this is just to begin inflammation in order to kill the bacteria. Now, C3b, we said it binds to bacterial membranes to create the MAC complexes, but they're actually also able to bind to antigen antibody complexes to do something called immune clearance. So C3b binds to antigen antibody complexes and binds those bacterial cells to red blood cells. So this one's kind of cool. I'm going to draw a red blood cell here. Looks kind of like a donut shape. We have the bacteria here. We have the antigen here. And we have the antibody here. And remember, when the antibody binds to the antigen, it slightly changes its shape to open up a binding site. Here you can bind C3b. And C3b is going to help bind the antigen antibody complex of the bacteria to the red blood cell. Now, why in the world would I want to attach the bacteria to the red blood cell? That just seems like a bad idea all in all. Now you're like attaching a pathogen to a red blood cell. Well, not quite such a bad idea, because where do we take these red blood cells to filter them and clean them? Well, we take all blood to the liver and the spleen. If all red blood cells have to go into the liver and the spleen, if you tag a bacteria onto it, this whole thing now has to go to the liver and the spleen to get filtered and to get cleaned. If your liver and spleen notice that there's a bacteria attached to the blood, the liver and spleen are going to do something called immune clearance. They're going to destroy the whole thing. So yes, you are going to destroy the red blood cell, but it's a give and take of, you know what, I can get rid of one red blood cell and I can get rid of the bacteria with it. Your body will make new red blood cells, so not to worry. The third mechanism is through phagocytosis, and you guys all know what this is. Macrophages are able to go and engulf cells. So the rule is that you cannot engulf a naked bacteria. If you just have a bacteria and you just have, let's just say, an antigen on it, this is considered a naked bacteria. Without the help of C3b, you will not be able to phagocytize it. So again, C3 protein breaks down into C3a and C3b. I've repeated now several times that C3b is able to bind to bacterial antigen antibody complexes here. So we can have a C3b binding here and C3b just binding all around the actual bacteria. When you coat a bacteria with C3b protein, this alone doesn't do anything except for send a signal. So this is my little signal lights around it. This C3b coated bacteria, we call this process opsonization. When you coat a bacteria with C3b, it's called opsonization. You've literally just made the bacteria look like a delicious meal for your macrophages. And the macrophage can now recognize and say, ooh, yummy, I'm going to eat this opsonized bacteria. So the C3b, its job is to coat the bacteria. It can create Mac complexes, and it can also do a process called opsonization, where it makes the bacteria visible and look tastier to phagocytes. The last one here, I just included the Mac complex. So the Mac complex starts with C3b, as you already know. We've repeated this now 5 or 6 times. C3b is going to begin the Mac complex. C3b causes activation of other complement proteins like C5, C6, C7, and most importantly, C9. You don't need to memorize any of the other complement proteins. Really, the only one that I gave you was C3, splitting up into C3a and C3b. Now, C3b, like we said, is going to activate other complement proteins. These complement proteins will come and they'll start lining themselves into like a circular orientation to create a pore in the membrane of the bacteria. So here are my complement proteins. And imagine if you had thousands of these in the membrane of the entire bacteria. That would cause the membrane to fall apart and the cell would rupture. That can cause cell death. So far we've been over the 3 activation mechanisms of complement, and we've been over the 4 destructive pathways of complement. Please make sure you know these, and if you are having trouble, please do not hesitate to contact me. So ultimately, once you have C3b activated in the presence of a pathogen, C3b is going to then downstream activate the other complement proteins to come and build this anchor-like structure, as well as the actual pore of the Mac attack complex. So these C9 proteins, they are what actually creates the pore. So although you don't need to memorize any of these proteins, what's important for you to know here is that C3b is what begins the cascade of activation of the other complement proteins in order to create the membrane attack complex. So this is where we'll stop this PowerPoint, and we'll pick up on this on the 4th and last installment of Lymph and Immune.